Yeah, so thank you for having me. I'm actually here as a researcher. I'm not in the clinical lab, but I am presenting on behalf of our technology development group, which kind of liaises between our research and clinical labs at the Institute for Genomic Medicine at Nationwide Children's Hospital. Okay, so if you're not familiar, gene fusions are often seen in pediatric tumors as the driver event. They can inform about diagnosis, prognosis, and there are targeted therapies for these events. These happen when there are chromosomal rearrangements and it puts two genes together, usually causes overexpression of one or both genes. So we are very interested in molecular assays that can identify those. On the right, I'm showing a graphic of in-house data. We've sequenced with RNA sequencing about 400 pediatric tumors to date, and this is kind of a schematic of what we see. Some known, some unknown, so we've built quite a database. So why are we using RNA-seq for fusion detection? Why not DNA sequencing? Great question. So the answer is that if you use genome or exome sequencing, there's little to no coverage of introns. And you can see here that when these gene fusion events occur, oftentimes the breakpoint can be in an intron, and you would not be able to detect that through DNA because, especially if you're using an exome capture, there will not be coverage of the introns. So RNA-seq, right, you won't have those introns, hopefully, and you can catch the exon-exon junction as seen here. So RNA-seq is a really good method to catch those hybrid transcripts. The genes that are often fused together in pediatric tumors are common in some subtypes of tumors, but the breakpoints are different. However, the exons that are captured together will be the same, like here BRAF. We often see the same exon, but different intron drug points. So RNA sequencing is a really good method to do this. Furthermore, if you are in a clinical lab and using targeted RNA panels, as we are currently, this also limits the ability to detect novel fusions because one of the partners has to be known. Okay, so I have one slide about informatics. We, back in 2021, put together a paper about our fusion detection pipeline. And this is really nothing novel. You can see on the left, I mean, we do RNA sequencing and we use a bunch of tools that have already been published and hopefully still supported. Sometimes they're published and not supported 3 years later, so we chose to go with 7. And basically, we use all 7 tools when we have RNA sequencing from a tumor. And we use an overlap script, and typically we look for at least 3 of the tools to call evidence of that fusion, and then we consider it real. That's the pipeline we put together. And you can just see on the right is a nice figure from, I think, figure 1 from the paper that shows some controls we use that I'll talk about soon from Seraseq, and that we were able to pick those up with our pipeline. So this is published if you're interested, and we still use it in our research lab. So we consider a lot of factors when we're evaluating RNA-seq kits. When we decided to test this watchmaker kit, this is not the first bake-off that we've done. There's always new things coming out. So these are some of the key criteria that we consider, some more important on the research side than clinical. But for this particular endeavor, we are hoping to validate this in our clinical lab. And so these were really some of the most important factors to us. The low input, of course, oftentimes we get fine needle biopsies, and that's all we have to work with. The timing was very important. So that was actually the first time I'd seen your slide. So I have the next slide. It actually does take 1 day, so I can attest to that. And the sensitivity and specificity of picking up tumors. We know that sometimes these are very lowly expressed in the RNA, which is kind of wild to think about because they're driving the tumor, but they're very lowly expressed, and we have to think about false negatives in that sense. So these are the criteria that we really consider to be of utmost importance during this bake-off. Okay, so one slide about our setup. We had 4 different technicians involved in this testing. Our positive control was Seraseq Fusion Mix V4, which has 16 known RNA gene fusions. And we tested patient tumor tissues. In addition to the control, we had patient tumor tissues that we had previously sequenced and validated in our CAP-CLIA space with known fusions. And these include both FFP and frozen. And we evaluated 4 RNA-seq kits. So we are already using NEB-NEXT and Takara. And we are looking again for something, as I mentioned on the previous slide, with a little bit more shorter labor and more sensitive to fusion detection. So hoping to add something to our suite of tools. And I will say the hands-on time, so if anyone here has used these kits, this is our hands-on time that we've determined from the SOP that we've written. So it might be different in another lab, might be different in the manufacturer-provided literature, but this is how long it takes us to do these kits. All right, so the objectives were pretty straightforward, as I've kind of already mentioned. So compare a new kit to our current in-house RNA-seq protocol. Again, this is all being done in the research space at this point. Test methods for lower input and FFPE, identify a workflow with shorter hands-on time, and always keeping in mind the end goal, which is validation in our clinical lab. Okay, so hopefully you can see this. I put forward, we look at a lot of metrics when we evaluate these kits, right? We don't just go straight to fusion detection. So we tried a variety of FFPE samples, frozen tumor samples, human reference RNA, which will be our negative control, and Sera-seq on the right, and the 3 different kits that I had mentioned, the NEB, Chi-seq, and Watchmaker. So in the top left, I'm showing the percentage of duplicate reads. This can be really dependent on your input, right? If you have lower input, you're probably gonna have more duplicates if you're using same PCR cycles. But also can indicate the robustness of the kit, right? How many of the molecules are really kept throughout and present in the final library. The average library size on the right and the bottom left percent of protein coding reads, something else we care about, was pretty comparable across all kits. And then another metric we look at is the percentage of ribosomal reads, which is on the bottom right. So some variability in those kits, but if you can see the scale, it's like less than 3%. So we don't care about ribosomal reads for fusion detection, so we want to minimize those, and this kit allows us to deplete that. All right, so next so this is a summary of like 2 years of work, but for low input testing, we considered low input to be 10 nanograms. I don't know what other labs are doing, but we really try to go in with 100 to 200 nanograms if we can. That would be considered our normal input, which is in the orange bar. And then low input, we consider 10 nanograms. Our extractions protocols are pretty robust, so 10 nanograms is low, but we can usually get that. So again, looking at the same metrics, the percentage of duplicate reads, the average library size, percent protein coding reads, and percent ribosomal, just showing a snapshot of both FFPE and frozen and controls here, but we really didn't see a huge difference when we did that. All right, so moving on to the actual fusion detection. So again, we use the Sera-seq V4 controls, and this has 16 known fusions, although that one at the bottom I don't think is known because none of our kits have ever picked it up. Sorry, Sera-seq. I don't know, it's just across the board. Maybe it's a limitation of the assay, I don't know. So here we're testing normal input, which is the first 2 columns, and low input, which is the right 2 columns. So we did see some discrepancies in what the kits were able to pick up. And then I'll just note at the bottom, I put the total number of reads. Obviously, if you have more reads, you might be more likely to pick it up. That was not the case with our low input testing. And we haven't answered this question yet, but we are targeting 100 million total reads for fusion detection. Typically for gene expression analyses, we target 80 million, and we think we're still getting more data and having a statistician look at it, but we think 100 million is good enough for detecting fusions. Okay, so this, again, we're using our OnFusion tool that I had presented in the beginning. This now we're actually looking at known fusions in actual patient samples, both frozen and FFPE, and I just put the technician up there. We didn't see any tech-to-tech variability, so we either have really good techs or the kit is consistent throughout. We do have good techs, I will say that. So known fusions, a lot of Kia BRAF, as you would expect in a pediatric lab. And the number of tools, again, we usually consider 3 tools to be good support for that, so probably we'll keep that consistent when we move through validation. And the number of reads of support on the right, just for curiosity. So we do know that Kia BRAF can be difficult to detect because there are a low number of reads. So again, we're still working on number of reads needed and to feel confident in picking that up. Okay, so the last thing I'll talk about, so not moving away from fusion detection, but complementing fusion detection with gene expression analyses, right? We're reading all of the RNA in this kit, so we have gene expression values. And we've typically used these on the research side. We've published a lot of this data, and we've shared it, which I'll show at the end. But we were very curious about how we can integrate this new data using Watchmaker with our previous data. That's something that's very important to us. We want to mitigate batch effects. We've already developed a lot of computational pipelines using other kits. So we were concerned, but optimistic about moving to a new kit and being able to integrate that. So on the left, I'm just showing 2 correlation plots between the NEB-NEXT and Watchmaker. Looks really great. Again, these are just every dot is a gene, and it's the expression value that we calculated from the same sample. We use human reference RNA on the top, and then the Sera-seq control RNA on the bottom. And then the other thing we do, again, in the research setting is display the tumor on some sort of reduced dimension plot. So here I'm showing a UMAP. This is our cohort of blood tumors only. They've all gone through RNA sequencing, and we prepped the same sample with 2 different kits shown, and they landed right by each other. So if you're not familiar with these plots, the tumors that have similar gene expression profiles will kind of naturally cluster together. So my guess is those 2 big clusters are myeloid versus lymphoid leukemias, if I had to guess, because I don't actually know. But so this is, it showed us that the kit can integrate well with our previous data. And also I'll just mention that we're not going to validate this initially, but these kinds of plots have actually helped us redefine diagnoses for some of the patients because they get a molecular diagnosis from pathology or whatever, and we actually see them land in a different spot on this type of plot. So it's been really helpful and useful data to have in our hands. All right, so again, on the theme of gene expression, so this is one slide from just a clinical case that we had, a case study, if you will. And so we have the ability, again, to go from research to clinical because we have the CAP-CLIA space. So this patient in particular was a female born with sarcoma. RNA-seq in research setting identified the NCHEK3 fusion. We clinically confirmed that with RT-PCR, and this allowed the patient to go on targeted therapy. What I'm showing on the right is a plot of gene expression of NCHEK3 only, so expression of that gene from the RNA-seq data. And I've binned it by different tumor types or subtypes, so you don't need to read them, but on the left is CNS and then medulloblastoma, et cetera. The red dot is the patient that we're looking at, and this is in non-CNS or extracranial solid tumor. And you can see it's pretty high expression of NCHEK3 compared to all other tumor types that are similar. So I, as a researcher, imagine wow, what if we only saw this plot on the right-hand side? Could that be enough to get this patient on targeted therapy? Because we know that fusions cannot always be detected through RNA sequencing, and there's limitations. So maybe that's a pipe dream, but we continue to generate this kind of data, and again, knowing that the Watchmaker data that we're moving to can integrate well with our existing database of hundreds of tumors makes us feel pretty good. So the next steps for us using the Watchmaker with Polaris depletion. So before we move to automation, which we are actively working on, and clinical validation, we have one more test to look at low input some more, and then test limits of tumor cellularity. So not every sample we get in our clinical space is 100% pure tumor. So we will perform some dilution studies to see kind of what is the cutoff for what we can accept for RNA sequencing for fusion detection. And my last slide, I just want to put a plug. So all of this data and analyses, especially the comparative gene expression analyses, could not be possible without our internal data, but also the Treehouse Childhood Cancer Initiative. If you haven't heard of it, it's based out of UCSC. And we formed a partnership with them in 2018. Here we are in the Redwood Forest. And their goal really is to crowdsource RNA-seq data from public repositories, from partner institutions like Nationwide Children's, and make it available to the public. And they run it through the same pipeline, so you don't have to consider batch effects and all of that. It's just ready to go. So you can visit their website for more information. It's freely available to the public. They have almost 3,000 pediatric tumor RNA-seq and over 11,000 adult. So I will end on that slide again. I am really here to present on behalf of our tech dev lab. They're really the brains and the hands behind all of this work. And all of this work is under the guidance of Elaine Martis, who is executive leader. Thank you.